that's a big deal. This is a randomized phase 3 trial of a novel agent, and it seems to be a positive trial. And in general, those types of agents get approved by regulatory authorities. When the phase 2 trial of this combination versus pembrolizumab was conducted previously, the hazard ratio was 0.51, I believe, for relapse, and it was like 0.41 for distant metastasis, meaning it reduced the risk of relapse by approximately 50%, and it reduced the risk of distant relapse by approximately 60%. If we're anywhere close to that, this is a blockbuster. If we're just, not quite as close to, not super close to that, but still obviously meeting the pre-specified threshold for benefit, which it met by the fact that they said it met its endpoint, it will be a significant reduction that will be clinically meaningful. So it's complicated, but essentially what you're getting is you're seeing what's different about the tumor, how does the immune system recognize what's different about the tumor, which of those differences are turned on, and then you calculate a prediction, you use a prediction model to say which of those differences should we target in a vaccine, and then you vaccinate. It's remarkably personalized. Most of personalized medicine, and I put in air quotes, is doing genetic analysis on the tumor and saying your tumor has this mutation. It may be a mutation that 40 or 50% of all tumors have, or it may be a mutation that 1 or 2% of all tumors have, but it's not personal. It's a mutation that does help to figure out, if you detect it, what that person should receive, but it's not like it's just unique to that individual. So the cool thing about this drug is not only does it appear to be associated with better efficacy, reduces the risk of recurrence more, it reduces the risk of distant metastasis more, but it doesn't make Keytruda more toxic. It just adds the inconvenient side effects 9 times of getting an injection. Yeah, it takes about a month. So we have to send in remember I said we do DNA sequencing and RNA sequencing. We need to send a sample of the tumor in. Then they isolate the DNA and RNA and they do a quality check. If it meets the quality check, then they can move forward and do the sequencing. And then they also do another check to say, do we have enough information to make a vaccine? And then the answer is yes or no. And then once they do that, then they can make the vaccine. So in some ways, our enthusiasm is warranted, but also should be tempered a little bit because we really haven't seen what the data looks like. We don't have those hazard ratios for relapse-free survival and distant metastasis-free survival. We don't really know what the comparative toxicity is. We just know what the one-liner says. The challenges ahead are twofold. One, there's a manufacturing challenge that needs to be sorted out. I know the companies are working on this, but it's not trivial to make thousands of vaccines that are personalized. Like when they were mass producing COVID vaccines, it didn't matter if I got your COVID vaccine or you got my COVID vaccine because it was the same. It matters if I get your vaccine and you get my vaccine. We got the wrong drug, right? So because it's an individualized drug, there has to be much more strict criteria in terms of making sure that the handling of samples is that we know what sample is whose, and then we know what vaccine, which is made based on the analysis of those samples, is whose, and then we continually are making it because you need to make 9 doses, and you need to make sure you ship it to the right place. All those things are logistical challenges that can be met, but it still needs to be developed.